Hello there! Have you ever wondered how living organisms reproduce and pass on their traits? The answer lies in a special type of cell division called meiosis. Meiosis is the process by which reproductive cells or gametes, which include sperm and egg cells, are formed. Unlike mitosis though, which creates identical cells for growth and repair, meiosis produces cells with half the number of chromosomes, ensuring genetic diversity in offspring. So the next time you see the amazing variety of life around you, just remember, it all started with meiosis. Here, the nuclear membrane is present, and the DNA exists in coiled form and duplicates, forming homologous chromosomes. With DNA replication complete, the cell is now ready for prophase 1. As you can see here, two sister chromatids have been formed, and through a process called synapsis, homologous chromosome pair up, forming a structure known as tetrad. Crossover also happens in the spot called chiasma. As prophase 1 progresses, the nuclear membrane disappears, allowing the chromosomes to move freely. In metaphase 1, spindle fibers attach to homologous chromosomes, preparing them for separation. The tetrads align along the middle of the cell, marking the official start of metaphase. This independent assortment ensures each gamete gets a unique mix of genetic material. In this phase, the sister chromatids remain together, but are separated and pulled toward opposite sides of the cell.
In telophase 1, we can see here that the separated homologous chromosomes reach opposite poles. Then, a nuclear membrane may start to reform around them. However, the chromosomes remain condensed, as the cell will soon undergo a second division. Now, in cytokinesis 1, the cytoplasm divides, forming two haploid cells. Each daughter cell contains one chromosome from each homologous pair, but these chromosomes still consists of two sister chromatids. Now, meiosis number 2 starts. In prophase number 2, this nuclear membrane breaks down again if it reformed. Then, the spindle fibers start forming and chromosomes recondense, preparing for another division. Unlike in meiosis number 1, there is no DNA replication before this stage. The next phase is metaphase 2. Here, the chromosomes still composed of sister chromatids align at the metaphase plate. Then, the spindle fibers attach to each chromatid's kinetochore, preparing to pull them apart.
Anaphase 2. In this stage, we see that the sister chromatids are finally separating. The spindle fibers, which have been attached to the centromeres, starts pulling them towards the opposite poles of the cell. Unlike anaphase 1, where the homologous chromosomes were separated, this time, each chromatid is moving away as an individual chromosomes. It's like a game of tug of war, except there's no turning back because the chromosomes must reach their destination at the end of the pole, or cell poles.
Telophase 2 is where the sister chromatids completely move to opposite poles and the nuclear membrane reforms. Cytokinesis follows, dividing the cytoplasms and forming 4 haploid daughter cells. Indeed, the genetic diversity of this process is crucial for evolution and survival, as it allows species to adapt to changing environments, reducing the risk of genetic disorders.